We're in chapter 4. No, we're in chapter 5. Because tomorrow we test chapter 4. Got to finish it. So I'm going to go to chapter 5 and break the seal and get into it.
We do the chapter 5 up though, did he? Okay.
That says not ready.
Yeah, we're still in mitosis.
We haven't finished lesson 4.
I'm sorry, we're testing lesson 3 tomorrow.
Yes.
Right.
I got it.
We just started
And I know I'm not here because it's radio mode powerful on my disc. So we're doing
Cell cycle.
there you go. Which one we do last? Y'all remember that?
I do. We started interface.
Can I ask you a question before you start, please?
Fire away.
Are we going to do 2 tests tomorrow or 1 test?
One test.
A lab as well?
Three.
Well, the lab's a separate class.
So we have 2 tests tomorrow.
Yeah.
Can I have my lab stuff back?
I don't have it.
So we have a lab test and a class test tomorrow?
We turned it in last week. We need that to study with.
We're having a class test. And then the lab class is a separate class. Like you going to English for a test there too. To me, to me they're different classes.
We have one teacher.
So the lab is going to be on.
So the lab is going to be over.
No,
I don't have.
what is that gonna be on?
We turned that back in.
Yeah.
And the whole shooting match. Let's see what I got ahead. Okay, let me 7. If I do 6 and 7, I'll be happy. Okay. A cell, well, I said, most of his life is spent just living, being a cell. And that's interphase. Because when that cell divides, that cell is gone. And there are 2 new cells where that cell was. In interphase, you got 3 subphases, G1, S, and G2. G1, he's just growing from a baby to an adult. He's just getting size, getting mass, he's getting bigger. S, he has word that right down the road we're going to do mitosis, get ready by making a double supply of DNA, enough for 2 new cells. So synthesis means he's synthesizing DNA. He's not synthesizing like ribosomes and reticulum, things like that. G2, he starts synthesizing the other things, the extra ribosomes, the extra mitochondria, the other things you're going to need to have when you divide yourself in half and have 2 cells. Okay? Now when G2 is over, of course, you enter mitosis. Now brain cells, they stay in here. They don't do this. They don't do those. They don't multiply. I mean, you hurt one, they don't heal back. Everyone you have right now, you're born with and you'll die with. They don't heal because they don't enter the S phase. They stay in G1 the whole time. They're still in G1 right now because they will not ever prepare themselves to divide. Because if you had a stroke, you could recover from it as you made new cells replace the ones that were hurt. You know it doesn't happen. And so, unfortunately, brain cells, they simply get off the cycle and they stay like they are for the rest of your life, yes.
Same would be with like cardiac.
Oh yeah,
cells
now
too.
some cardiac cells can heal. Depends on the degree of damage done to them. A minor heart attack, you'll see scarring, but you'll see healing also. But the massive heart attacks, you rarely recover enough to make it much longer in your life. That's when you become a candidate for a new heart. Okay? But the cells that are notorious for getting off the cycle are the brain cells. But they don't keep on making more of themselves, unfortunately. G1 is a long time. It's a long time between G1, between mitosis and the S phase, a long time that sucker just growing along. And like I told you, in the S phase, you're really concerned about synthesizing the DNA. So you have enough to make for 2 new cells. So he grows protein, meat. He just takes, and G1, he just growing, doing what cells do, growing. Everything is dividing and being replicated except for DNA. And it's so complicated, the cell waits and does it at one time during the S phase. Now, the chromosomes in G, they don't have, you only have 46 chromosomes in G1. That's what you came out of mitosis with. Okay, so they stay there. But you can't see them. They're like long strings of fishing line all balled up. You see a ball of stuff. You go, how many little segments are there? You can't count them. If I gave you like 6-pound fishing line and cut it into 3-inch segments and put them all together, water them all up, you couldn't count those things till there are 100 of them, unless you pull them out one at a time. And in a way, as the body thickens them, I said, oh, there's one right here. See how thick he is? And so as you thicken them, the chromatin becomes chromosome. And when you can see it, we call it chromosome. And when it lengthens back out, stretches back out again, and you can't see it, the name changes right back to chromatin. Chromatin, chromosome, same thing. One's invisible, one's visible. They both got DNA. All right. So during G1, the nucleus is there in all its glory. It's got the membrane, everything's fine. The nucleolus is, you can see the little dot inside the nucleus perfectly. There's some pictures now. When my wife first saw these, she thought these were cells, but 2 nuclei. Why was she wrong?
Oh,
Here is your cell. Here is your nucleus, and what is that? It's possible for a nucleus to have 2 nuclei. So here again is the cell. There's the one nucleus with its 2 nuclei. Well, there's one nucleus there. There's 2 nuclei. There's one nucleus. But when she first saw it, she likes to kind of quiz me, like she'd know I'd miss one. But I said, no, that's the nucleus. And then she saw, oh, I see now. But that's a good one right there. That shows you clearly that there's your cell boundary. And there's the nucleus and this light area inside. Usually it's dark, but that area is the part of the nucleus that makes your ribosomes.
Can you have more than 2?
You can't. And sometimes you have more than 2. But all the ribosomes you find out here are being made right there and spit out into the cytoplasm. Ribosomes don't make any ribosomes. The nucleolus makes the ribosomes. We didn't know that for a long time. For a long time we said, well, there's the nucleolus and don't know what it does, but there he is. And I don't know what he does. He's making ribosomes. He's full of RNA because ribosomes are made of RNA. Okay? Now after you leave G1 and you've gotten pretty big now and you've been growing, you enter the S phase, and that's where it's time to make copies of your DNA, because one down the road you're going to need a double supply for the 2 new cells. And if one cell's got 46, I better have 92 on hand when I divide myself in half, right? So at some point during this G2 phase, that bad boy he's 4N. Now what I mean is this. Your regular number is 46, right? That's 2N. What's half of 46?
23.
33.
23. That's 1N. If N equals 23, what's 2N going to be?
46.
46. Now, when I double 2N become 4N, what is 4N going to be? The 92. What happens, I can divide 4N and say, hey, you bunch go this way, you go this way, and I'm right back to my regular 2N number. 2N is regular. You won't 2N. Because 1N came from mama and 1N came from daddy. So in my cells now, of all my DNA, half of it, 23, is from my mother. The other half, 23, is from my daddy. When they're both together, we say that's 2N. It's just mama, well, that's just N, and daddy is the other N. So 23 to 23, that's 2N. That's how that works. And a lot of folks don't understand. That's called ploidy of a cell. We discuss haploid and diploid and tetraploid. And that's what we're talking about. Tetra means what number?
3.
4. So 4N would be tetraploid. 2 N's would be diploid. And one N is monoploid, exactly. That's how the numbers work. And when I say 2N, if you're a piece of corn, you reckon your N's going to be 23 or some other number? Some other number. So I don't need numbers to discuss how these chromosomes divide themselves up. I know you have half and now you have twice as much. All right? After he's finished, and he now has the tetraploid number, he has a lot more he's supposed to have because he's getting ready to divide. So when you're in G2 phase, you really have 92 chromosomes. But you're just using your original ones to control your cell activities. The other 46 are designed to go to the other cell when you divide yourself. Okay? The organelles, ribosomes, if I have 100, I need 200 for my two new cells. If I have 1 foot of ER, I will need 2 feet, 1 for this cell. So in G2, you're making more of the stuff you got to have when you divide yourself in half. So that's just you getting ready. So S, you're getting ready to divide by making more DNA. G2, you're ready to divide by making more organelles. In G1, you're not doing anything that's growing. You're still so little. Okay, now, I'm not going to go any further because regulation is a whole new ball of wax.
We'll go back to 7.
Go back to which one?
221.
There you go.